the most important fruits grown around the world. This is due to its popularity as a multi-purpose fruit, not only as a dessert fruit but also a source of chemical compounds used in medicine, such as popping, camomile, and carpine. The year-round availability of papaya makes it an excellent source of vitamins and minerals. Minerals like magnesium, potassium, and vitamin B, like pantothenic acid and folate, abound in this food, making it a potent antioxidant powerhouse. For the first research background, some of the main groups of phytochemicals that have been suggested as natural sources of antioxidant are polyphenols, carotenoids, vitamin C, and vitamin E. All of these may contribute to the antioxidant activity of plant matter as a whole. So the second one is, several studies have shown that phenolic compounds are the most important bioactive plant chemicals that are good for human health. In fact, many authors have found a direct link between the total phenolic content of seeds, fruits, and vegetables and their antioxidant activity. And for the last one, the Caricaceae family is where the carica papaya comes from. Several species of the Caricaceae family are used to treat a wide range of diseases. Papaya is a steady plant that was first found in the southern part of Mexico. For the first problem statement, into different parts of the papaya, which parts of the papaya have the potential to be transformed into added value in the product as a health benefit for the body. The action of radical scavenging poses a significant threat to our body as well as our health. And for the second problem statement is the parts of the papaya known as the flesh of the papaya and the peel of the papaya. Both contain a vitamin and antioxidant. However, it is unknown whether the amount of these substances vary between the two parts of the papaya or which of the two parts contain the most antioxidant overall.
Research objective. The study focused on the development of a strategy to assist developers and researchers. Number 1. To find out which components of papaya has the highest antioxidant content. Number 2. To find out how much antioxidant activity each part of the papaya has. Number 3. To figure out the total amount of phenolics in the parts of papaya, which are the peels and the flesh. Research question. What is the total amount of phenolics content in the different parts of the papaya, such as the flesh of the papaya and the peel of the papaya? The next research question is how many total antioxidants can be found in papaya? Number 3 is which part of the papaya has the highest total phenolic content as well as the highest total antioxidant activity?
For research scope, it is to precisely quantify the total phenolic content as well as the antioxidant capacity of papaya peels and flesh. The second research scope is conducting an evaluation on each portion of ripe papaya so they can determine the total amount of phenolic content and antioxidants that are present in each part of ripe papaya.
Significance of study. The papaya medical characteristic. It was traditionally utilized as a form of treatment for a variety of diseases. 2 is juicy flesh and peels. Both have a lot of nutritional value.
Our operational definition. Firstly, ripe papaya. As the papaya start to ripen, its skin will slowly start to change from green to yellow. The papaya is ready to eat when it almost completely yellow. They slightly squishy to the touch. Secondly, antioxidant, a substance that stops oxidation, especially one that protects cell from the sometimes harmful effect of oxidation like vitamin E, vitamin C or beta-carotene. The carotenoids and notable lycopene are beneficial antioxidants found in papaya that can neutralize the free radical. UV spectrometer. The spectrometry is a method for the figuring out how the many molecules are is something based on how much the light is absorbed. Different color compound is a technique used to measure the light absorbed across the ultraviolet and visible range of the electromagnetic spectrum. Next, the total phenolic content. Important plant part called Phenolic compounds have the redox properties that gives them the antioxidant activity. The typically phenolic compound with antioxidant action include the most phenolic acid flavonoid and phenolic acid are most abundant family of phenolic compound, notably in fruit and vegetables.
Expected outcome. Number 1. We expect to find out which part of the papaya has the most antioxidants. Number 2. The overall amount of phenolics in the peels and flesh of papaya should be expected.
This is the framework for our project. We utilize a UV spectrometer and DPPH analysis to analyze the antioxidant content of papaya peel and flesh as our raw material to determine which papaya compound contain the most antioxidant. We used the papaya peel and papaya flesh for this analysis. For the analysis, we use the material and chemical. We utilize methanol and DPPH to obtain DPPH analysis. Okay, for the next chemical and material to ascertain total phenolic content, we use Folin Ceotelis reagent and sodium carbonate solution.
Methodology. Sample preparation. The first step is the flesh and peels of ripe papaya are washed and sliced in 1/2 inches. The second step is the sample will put on tray and placed into drying oven at 35 Celsius for 16 to 18 hours for drying process. The third step, after the drying process has been done, the samples were powdered using cyclone mill twister. The next step is the process of extract sample for 2 parts of papaya was 1.5 gram of the peels and flesh of papaya are weighed using the analytical balance. The next step is for the extraction method. 1.5 gram of salt sample peels or flesh were mixed with 5 ml of ethanol in distilled water, and distilled water was added to make 100 ml. The final step is the volumetric flask that was contained. Extraction of sample is covered with aluminium foil. The covered volumetric flask is stored in chiller at -27 degrees for 1 night for further analysis.
So for the methodology, the procedure to carry out total phenolic content analysis is first, each of samples extraction, which is flesh and peels, were filtered using filter paper wetman, number 1. Then 0.5 ml of extracted sample was added to distilled water to make 10 ml in a volumetric flask. Third, the sample in test tube was stored in that place for 30 minutes. In the last one, in test tube, 1 ml was pipetted and added into 2 ml of Folin-Ceotelis reagent and 2 ml diluted sodium carbonate, which is 4.5 grams sodium carbonate that was diluted with 60 ml distilled water.
From the data analysis, results were expressed for average into 2 sample because of the duplicate sample. Use the Microsoft Excel to make the data and graph of the result for the total phenolic content and the total antioxidant activity.
For this Gantt chart, the content that we will carry out from week 1 until week 14 is briefing and discuss with our supervisor, sample preparation, carry out analysis of total phenolic content, carry out analysis of DPPH method, record data and thesis writing. We'll also do presentation to see the progress, and the last one is final report thesis writing.